AGRF does proteomics now. So it does this for a number of reasons, or AGRF has chosen to pursue proteomics for a number of reasons. The most obvious being that we're in the age of multi-omics, and so it's really a strength to AGRF as an organization to be able to provide multi-omic analysis to its clients. But also with a specific reference to proteomics, the field of proteomics is in a very interesting stage at the moment. There are a number of technologies which are entering the market and being widely adopted by researchers, to some extent clinicians, and in industry settings as well, which utilize existing genomics and transcriptomics expertise and workflows to analyze proteins. And so you can appreciate that AGRF, with its exceptional expertise in those genomics and transcriptomic fields, these sorts of new proteomic analyses really do fit in well with our existing expertise. But I think one of these technologies being, of course, Olink, which this the topic of this seminar. But I think in order to understand really the strong use case of Olink in the context of broader proteomics, I really want to take a step back and discuss proteomics as a field in general and the types of technologies which are really commonly present in proteomics literature and in proteomic studies themselves. So we'll get into it. I, of course, echo the sentiments previously by Saurabh regarding the traditional owners. So the human genome predicts roughly 20,000 or so proteins. However, the human proteome is far more diverse than that when you consider the isoforms and post-translational modifications that sort of fall out from any one translated protein. There's really a great diversity of protein content within not only humans, but also across species. And it really sort of highlights the necessity of broad strokes proteomic analyses. Proteomics is, of course, the study of these proteins. It's an established field. It has really quite an interesting history here in Australia, particularly, but also has a number of these emerging technologies. And I've sort of given a brief rundown of some of the existing technologies within proteomics, and with specific reference to the types of information that they can provide. So, for instance, proteins being these really diverse three-dimensional structures, it's terrific to understand those three-dimensional structures themselves. And so you do that through structural proteomics with things like X-ray crystallography. There is, of course, the sequence, the underlying amino acid sequence, which is terrific information, particularly in other proteomic workflows in terms of identifying specific proteins. These are one of the earliest forms of protein analysis in terms of Edman degradation to fundamentally understand protein sequences. And then you also have functional proteomics, because proteins are the functional molecules of biology, more or less. And so understanding the way that one protein or a bunch of proteins interact with each other and with other substrates underlines functional proteomics, and you can analyze that through things like co-immunoprecipitation studies and kinematic studies and PPI networks, all of which sort of have a strong presence and growing presence within the literature. But I want to focus more or less and dig down deeper into expression proteomics, which I believe sort of provides a really good fundamental base level for a proteomic experiment or proteomic project and proteomic analysis of a wider biological context. And this is done by a number of technologies, which we'll discuss further in the preceding slides, including immunoaffinity assays, which I'm pretty confident people in this room have experience with, mass spectrometry, which I'll discuss further, and also proximity extension assays, which are almost the new kids on the block when it comes to high-throughput proteomics. And sort of the commonly touted reasons why protein analysis is fundamentally important for a project is that ultimately, as I mentioned, proteins are the functional molecules. They're the things which drive the biology within biological states. They're also druggable targets, so you can appreciate there is a significant degree of translational significance there, I suppose. And they're also biomarkers of disease, which we'll hear about further in the next presentation. And I think a question, as I mentioned before, expression proteomics provides this foundation from which we can build all of these other proteomic studies upon. A fair question which often gets asked by people is, if we're looking at expression, why not just look at transcriptomics? Transcripts are often poor surrogates for protein levels. It's not just me saying that, it's all of these people here as well. And they've been, as you'll note by the dates, they've been saying it for quite some time. So really there isn't a one-to-one surrogate for looking at the proteins themselves. They are really a fundamental element of biology, and it should be they should be assessed. So to delve deeper into sort of the existing technologies which drive or which are used in expression proteomics projects, um, immunoaffinity proteomics is probably, as an undergraduate, you get exposed to this almost straight away if you're doing a molecular biology or biochemistry degree. And really, they're defined by the use of antibodies as affinity agents to bind to specific proteins. Examples include immunohistochemistry, which has been existing in some form or another since the 1940s, where you probe basically a tissue layer. ELISAs or enzyme-linked immunosorbent assays, which have been around since the 1970s. This is sort of a more liquid assay itself. And then Western blotting, which is a sore spot for me, considering how much I struggled with it during my PhD. But those are ones where you just embed your proteins on a bit of nitrocellulose paper and probe them with antibodies. And then more recently, I say recent, past 20 years, 25 years or so, we have more high throughput methods in the form of antibody arrays, where the actual assays are conducted either on chips, in microdotted plates, or on beads. As I mentioned, these are thoroughly adopted by the research and medical communities. There are IVDs existing which have ELISAs as fundamental parts of the workflow. But there are fundamental issues with these immunoaffinity assays as a proteomic method. One being that it restricts proteome coverage in the sense that you're not going to see more proteins than you're assaying. So if you have, if you probe 2 proteins on a Western blot, you're not going to see any more than those 2 proteins. It's sort of logical. Consequently, there's traditionally a lower throughput, both in the samples that you can analyze. There's only 12 or 15 lanes in a gel, and then the numbers of proteins that you can analyze at any one point. And also, not to throw shade on any vendor of antibodies, but some vendors don't provide the best antibodies. I should know, again, my PhD. However, overwhelmingly, the most commonly used technology platform within modern proteomics is mass spectrometry. And this is, it's often touted as a true proteomic technologies. I think this is unnecessarily divisive, but it's a very attractive platform for a number of reasons, one of which being its compatibility with many sample types. You can pretty much rock up to a mass spec with a protein sample from any patient, any species or anything like that. So long as you have a reference proteome to compare against, you should be able to get some protein IDs out of it. It has a terrific suitability for discovery proteomics in the sense that you don't know pretty much what you're going to find at the end of a proteomics experiment with mass spectrometry. It's very much probability based in that sense. And then also has a qualitative and quantitative proteome coverage in the sense that it tells you what's there and can tell you how much is there. Similarly to some of the other methods, mass spec has really quite a long history. The initial technology was established in the early 20th century, but was more refined in the 80s and 90s as a consequence of advances in sample ionization, which I'll get to in a moment. But yeah, so the modern state of proteomics is sort of heavily invested in mass spectrometry as a platform technology, except for a couple of instances where we'll talk about in a moment. Oftentimes, when I talk to people about mass spec, they often recoil in either fear or disgust, which is fair enough because these aren't particularly simple instruments. They're very difficult to understand, and the outputs that they have are often difficult to understand unless you have a significant degree of bioinformatic experience. But really, at a fundamental level, all they are is just a very accurate way of measuring mass. And the way that they measure mass depends on the type of mass analyzer that they have. And also, there are a number of mass analyzers which exist on the market which determine the type of mass spectrometer. So if you walk through a mass spec facility, good fair chance you'll see some of these. So a quadrupole, which is this one right here, measures mass based on the path of a particular molecule through 4 electrode poles. There's an Orbitrap, a really heavily utilized one, which analyzes mass based on the frequency of rotation around a barrel. And there's also a time of flight tube or a TOF tube, which basically measures mass based on how quickly a molecule passes through space. You can combine these mass spectrometers together, so have multiple mass spectrometers into a singular instrument where you get tandem mass spectrometry. So you can have a TOF, you can have a quadrupole leading to a TOF, which gives you a QTOF. But you can also attach a liquid chromatography unit. And putting all of that together gives you, in my opinion, what's the most commonly used proteomic technique or technology, which is liquid chromatography tandem mass spectrometry. It's a robust workflow. It's popular for a reason, but there are a number of caveats which come with it. One of which being they're not cheap. These things run, a couple $1 million for some of the high-end units. And you can get around that by going to platform facilities, I understand. But nonetheless, you can't get around the fact that substantial sample preparation is required. You can't just rock up with your tissue culture supernate and say, hey, give me proteins. You do have to do a series of digestion steps and reduction and alkylation steps in order to get fundamental protein readings out of it. And there's a really sort of a technical element of the workflow which limits its utility in specific sample types. This technical element is what's known as ion suppression, and I don't want to get bogged down too much, but basically in order for a mass spectrometer to work well, the samples need to be ionized. And the efficiency with which the mass spectrometer can ionize these samples is directly related to how well your data looks at the end of it, basically. This ion suppression is commonly observed when analyzing multiple compounds in an individual sample, when analyzing samples with disproportionately abundant proteins, and also in samples where there are large charged molecules. And it just so happens that there is a very popular matrix or sample type where all of these things come together in one sample type. That's plasma and serum. Traditionally, mass spectrometry has been really not the best workflow to use for plasma serum proteomics. You can appreciate this is the case because there's thousands of proteins in sera, not only at a basal level, but also in disease states where they're secreted into the plasma and serum. And there's also only 20 proteins that make up 99% of the protein content of plasma and serum. There is this vast dynamic range in plasma and serum proteome. And secondary to this, it also contains phospholipids, ions, and xenobiotics, which can interfere at some degree. And so this is really where proximity extension assays, and by extension, Olink, step in the sense that it overcomes this issue of ion suppression, and it overcomes the issues of mass spectrometry as a platform by using very large panels of affinity reagents. In that sense, you can do a terrific discovery proteomics without needing to deal with the ion suppression of mass spectrometry. The way that it works is that you have complementary oligonucleotides, which are attached to a pair of affinity agents. Depending on the vendor, the affinity agents may change, among other things. But in the instance of Olink, those affinity agents are antibodies. The antibodies bind to the target protein. The oligos are brought in close proximity and hybridize. And then this can be extended in an index attached, which tells you the sample type, the sample and the protein itself. Expanded using PCR, and then depending on the method, you can have a qPCR readout or an NGS readout to give you a quantitative reading of the proteome, where basically the number of counts of index that you have is proportional to the amount of protein of any one particular identity there. As I mentioned, there's a couple of vendors out here, the main being Olink, which we are, of course, service providers for. We're very proud to be a service provider for Olink and offer quite a diverse range of proteomic workflows for the particular project that you might be interested in. I'll give a brief rundown of what they are here. Explore HT. This is basically the largest proteome offering. It assays 5,416 proteins. These proteins are of diverse biological significance and origin. And so really it does give you a terrific proteome coverage of the particular biological state that you're interested in. And you can build really quite a robust differential expression analyses from the data which comes from the Explore HT workflow. There's also the previous generation Explore, which is 8 biologically themed panels of around 370 proteins, depending on the panel. These are themed according to biological themed according to biological themes, being inflammation, cardiometabolic, neurology, and oncology. And so you can appreciate that these would be really useful in an instance where you have an idea of, or you know what disease you're interested in, or the rough biological theme you're interested in, but you're not necessarily sure of the specific proteins you're interested in. So in that sense, it's kind of like targeted discovery proteomics. There is also Target 96, which is a far more targeted approach in that it assays 92 proteins. And there are a large number of biologically themed panels in this, similar to the Explore workflow. However, I can provide you with a list if you're interested in those. There is also Target 48. Target 48 is an important one to talk about because it's the first panel which gives you an absolute quantitative readout. That is to say, the data that you get at the end of the workflow is a picogram per mil reading. The previous workflows, being Target 96 and the Explore workflows, give you a relative quantitative readout. That is to say, a normalized protein expression value from the results. I'll discuss a little bit, the types of analysis which can be built from that. But Target 48, if you're looking for absolute quantitation, that's pretty much the way you go. There are also a couple of custom options. They do come with a couple of caveats. So if you are interested in custom panels, please come talk to myself or Brenton, who's in the audience here. As I mentioned, Olink is pretty much purpose-built for plasma and serum proteomics. However, there are alternate protocols which exist for a number of sample types, and there is published utility in a number of different sample types, including tissue cell lysates, conditioned culture media, peritoneal fluid, CSF, and a bunch more. So it's not the case that whilst the greatest use case exists within plasma serum proteomics, there is broad utility in a great many sample types. There's also a new product that was announced, I think, earlier in January. I should know because I attended the 1:30 a.m. stream from my bedroom. So this Olink Reveal, it's a highly curated panel, and it sits in terms of proteome coverage, I suppose it's 1,034 proteins, so it can kind of sit in between the targeted workflows and the Explore panel. It has high inflammatory protein and immune response pathway proteome coverage, but it's heavily curated. It's heavily curated in the sense that it covers 64% of all reactome pathways and 100% of all top reactome pathways. So you can appreciate from a bioinformatic perspective, this is really still a very useful tool when doing discovery proteomics and wanting to build a robust differential expression analysis. As I mentioned, it's validated in plasma and serum, but it's compatible with alternate matrices, though, as you can appreciate, because it's a new product, there's yet to be papers in this field. In terms of the specific outputs of the Olink service from AGRF, except for some of the custom panels and Target 48, the output you'll get is relative quantitation, the normalized protein expression value, which basically tells you which sample, condition, or treatment, or whatever you're looking at, has more of protein X, not necessarily how much protein X is there in sample, and also how is there more of protein X than protein Y. Having said that, this is still a very common form of data delivery in terms of proteomics workflows in general, specifically high-throughput proteomics, and is still something from which you can build a differential expression analysis. So after we do a little bit of QC, as I mentioned, we pretty much just deliver the data in a form of CSV file with these normalized protein expression values. The NPX values, if you're interested, is just a log2 scale with expression relative to an internal control, which is run in the assay. And this is delivered as a CSV file, as I said, which is 100 megabytes in size, which apparently my bioinformatics help says that this is very small compared to existing sequencing outputs, which is terrific. In terms of the actual analysis of the NPX values, you have a number of options, both free and internal from AGRF. We have a terrific bioinformatics team, chief among which is Matt Andrews, who's not here, unfortunately, but he is exceptional at differential expression analysis, exceptional bioinformatician. You can also analyze the raw data through a free browser-based tool called Olink Insight and do sort of basic QC in terms of PCA plots and basic differential expression in terms of t-tests. If you are highly skilled in R or skilled in R, I'm very jealous. You can also use Olink Analyze for more robust statistical tests like the use of linear models. There's also a number of technical considerations that we can discuss if I don't want to sort of dwell too much on this, but we can discuss the technical considerations specific to your projects. But broadly, I should mention that the antibody panels target human proteins. There are two exceptions in the sense that there's a Target 48 and a Target 96 panel, which targets mouse proteins. But the broad proteomic offering and proteomic analytical offering from Olink is in human proteins, or there has been some utility in non-human primate animal models, but these are antibodies which are directed towards human proteins. In terms of submitting samples to us, they can be shipped on dry ice. You really want to keep freeze thaws to a minimum, and you really want to keep pre-analytical variability to a minimum, and not only with Olink proteomics, but proteomics in general. 40 to 50 microliters is typically what we request. However, if volume is an issue, we can go as low as 20. Study design is important. Again, these are sort of, if you have a study that you're interested in running Olink on, please by all means do come speak to us, and we'd love to hear about it. And specifically as well, if you do want to if you do want AGRF to perform the bioinformatics, it is important that you tell us what comparisons you're actually interested in, and if there's any significant covariates and types of providing enough information about the samples such that comparisons can be made. Finally, there's a requirement for bridge samples across continuous studies. We can go through this further if you do have a study plan in mind, but this is to say if you're running kits, if you're running samples across multiple kit batch lots, there is the necessity for bridging. And finally, I just wanted to mention that with some of these technologies, it's often easy to view them as sort of a flash in the pan fad. I don't think at all that that's the case with Olink, and the adoption is quite significant, not only by specifically the proteomics community, but also by the broader research community. And it's leading to really quite high impact publications, not only in the large journals, which everyone wants to get into, like Nature and Science, but also in the very smaller, technically minded journals like JProRes. So I think there's a really significant interest across a very large section of the research community to pursue proteomics with Olink, and by extension, AGRF. And with that, thank you for your time.
I'm Clarissa. I'm the CEO and founder of Ketim Technologies. We're a Brisbane-based company, launched only last year in January. So we're just a little bit over a year at the moment. And we've got some really interesting results to show from our pilot study that we did in our first proof of concept study, in partnership and in collaboration with AGRF as well as Olink. So I really would like to just talk to you a little bit about the translational aspect of biomarkers and proteomics. We've heard a lot about the technical background of proteomics, and I guess from my point of view, it's how can we apply this for impact? How can we apply what we know and what we've seen in research into a clinical output, something that would allow us to give back to society as a whole. And that has always been my passion and my goal. It isn't just about discovering new things. That is the exciting bit. But how can we relate this back to clinical health, and how can we relate this back to delivering something that's impactful for people? So I'd like to discuss a little bit about precision medicine for mental health. That is my background. So I do have an extensive background in molecular development and assay development, as well as biomarkers and molecular biology. But my real passion is mental health. And I did my PhD at Queensland Brain Institute, where I actually was working in collaboration with the Mayo Clinic. And I was looking at discovering and seeing if we could understand predictors to antidepressant response. But post that, I really just wanted to kind of give you guys another insight of where else we could apply this. Now, if we talk about mental health, we know that an estimated 2 in 5, so 43% of people aged 16 to 85 experience a mental health disorder in their lifetime. This is data that's been recorded from the AIHW, the Australian Institute of Health and Welfare, and you can see over here that it equates to about 8.5 million Australians. Now, when talking about a mental disorder, we're talking about all mental disorders, so anything that has been reported to their GPs or have been self-reported. And majority of the mental disorders are really depression and anxiety. So we have a huge prevalence and really we're in a mental health epidemic at the moment, where we're getting a lot more diagnosis and people who are self-reporting their mental illness. I think what was striking for me when I came across this graph, if you see over here, there's types of serious illnesses by age and sex from 2009 to 2021. And if you look over here, there's the total amount and then the total age group. And you can see that the serious illnesses here, and look at this arrow here where this orange line is basically increasing from, just from 2009 up to 2021. That is roughly almost double the rates that we're seeing today. And this number isn't decreasing, it isn't stabilizing, dropping. In fact, it's increasing, especially post-pandemic. We are seeing a lot more cases. And the sad bit is that we're seeing a lot of this in the younger age groups. So the high risk is really in that 15 to 30 age group. And, it's really critical that we address this. But I think what I'm passionate about is that you can see over here that postnatal depression, which is the area I'm going to be talking about, is included under the umbrella of depression. And if we look at that fact, do you see these numbers are very similar in postnatal depression as well? So postnatal depression actually affects 1 in 5 women. And we know that as well, that there's a lot of mechanisms that, cause or biological factors, underpinnings, underlying depression as a whole. There's health behaviors, there are biological factors, there's pharmacological effects, and, all kinds of different causes and genetics and shared environmental risk factors. So we know from the research perspective as well that mental disorders are clearly a very heterogeneous and highly interrelated disease. So how can we start to identify or pinpoint what's happening in mental disorders when it's so heterogeneous? And I think that's where the beauty of biomarkers come in, because not only can we assess and use biomarkers as a tool to screen, but we can also use it as a tool to maybe start looking at subtypes and different areas of mental disorders, whether we can start to classify them instead of going through the regular self-reporting questionnaires that we use today. So in the Diagnostic and Statistical Manual for Mental Disorders in the DSM, postpartum depression is actually classified as a major depressive episode within the first 4 weeks to up to a year of post-birth. And when you talk about how we're looking at mental disorders today, everything is really based upon that initial questionnaire that you get from a GP, whether it's a self-report questionnaire. The problem with this is that we have this really subjective and, almost biased nature of not knowing how, somebody really feels. It's just based on their ability to recollect their own kind of feelings. So part of what I did when part of my story is that when I gave birth to my second child here, I was actually diagnosed with postpartum depression. And I think that was really quite striking to me because when I had my first child, I believe I had it as well. But coming from that point of view, it's like, this is just my first child. This is probably quite normal. You kind of have that self-doubt where you think, I'm sure this is not really anything. And I think this is the core fundamental issue here, that there's still a little bit of that stigma attached to mental disorders, where if you were to say you're depressed or you're anxious or anything like that, it might reflect badly upon you. But part of my goal was really to advocate and to destigmatize what we see in mental disorders. So I volunteered for the Perinatal Anxiety and Depression Australia. And as part of my champion volunteering there, I was interviewed by 9 News on my story to basically spread awareness about what postpartum depression is about. So really, I came to this conclusion where I have this desire to help others who are also suffering. I was, lucky in the sense that I was assessed pretty early. I was put onto antidepressants pretty early, and I responded to it really well. And it gave me this kind of strong desire to want to see how can I actually understand the neurobiological mechanisms under these mental disorders, and can I actually do something about it? So that kind of led me to this discovery kind of path where I then, decided to do my PhD to understand the neurobiology. And past my PhD, I really wanted to change and challenge the current beliefs and biases that we have in mental disorders. So using my own experience, my own personal experience, as well as my scientific experience, I'm really just going from my postpartum depression to a PhD to a startup. So as you can see over here, my kids are really important to me. I've got 3 now, and maybe you guys might be wondering how the heck I do this with 3 kids. But really, it's because I think I have just that real passion and desire to help others. And I think, I've got so much good support around me as well, including AGRF and Olink coming on really early to help me on my journey as well to discovery. So if you look at depression as a whole, when you're talking about the underlying mechanisms of depression, there's really many different, areas that you can concentrate on. You can look at the molecular kind of area, the molecules and down to DNA, genes, proteins, or you can look at the macro level, so looking at circuits in your brain, looking at fMRIs and brain imaging scans and things like that. Of course, you can look at behavior in animals and clinical trials. And a lot of it is really got to do with understanding the susceptibility and the resilience, cognitive flexibility, stress and coping, as well as neuroplasticity. So my goal is really to understand how does these concepts interrelate with the molecules and the biological mechanisms, and how can we then apply it to in a clinical setting. So you can see over here, I've touched on this a little bit as well, but it's really being able to understand, I guess, coming from that molecular point of view, is really going back to the core. But it's not just staying there. It's also understanding how we can apply this to cells and to animals and to large systems, and finally going on to behavior. And this is the central kind of dogma, I guess, of neuroscience research, that you go from what you see at the micro level to the macro level. But I think what's missing from this is, how do we apply it? How do we translate it? How do we get this to clinic? And that's kind of what I want to chat about. So when I set up CATIM Technologies, I really asked this one question: can biomarkers help in diagnosing postpartum depression? As I mentioned to you earlier, there is a real problem with postpartum depression. We know it affects 1 in 5 women, and yet there's still no objective test today to diagnose or to predict postpartum depression. So we have this problem of having a high number of people who are undiagnosed, misdiagnosed, and getting a very poor treatment plan that isn't based upon a personalized approach. And the problem we see, with this today is because we still rely on very biased self-reporting tests. So we know the current approach to healthcare in mental disorders, and not just for postpartum depression, is that you're pregnant, you give birth, and then 6 weeks later, you asked to maybe do a EPDS, or an Edinburgh Postnatal Depression Scale. And that's a universal tool that is used across the world. And it's a 10 questionnaire, and you basically get told, you do this based upon your thoughts and your feelings in the last couple of weeks. I don't know about you guys, but I can't even remember how I felt yesterday, let alone if I have a newborn that's been keeping me awake all night. I do have a 15-month-old, and it still feels very fresh in my memory. But, having to do this test that isn't quite so objective. And then if you're bad enough, you might be given medication. And this medication, again, would be a one-fits-all approach. So you might be given antidepressants. You might have no effect to it. You might have a negative effect to it. And you might have a really good and strong positive effect, like I did. But the research shows that even when you're taking antidepressants, about 30% of people, not just moms, but 30% of people on antidepressants will fail to respond even after multiple trials of antidepressants. So I came up with a solution. I thought, hey, can we look at bloods? Can we actually identify if there are biomarkers within the blood? Can we then use advanced analytics and multi-omics approaches to be able to come up with an individual risk profile for postpartum depression? And based on that, we can start to intervene really early. So we're looking at something that's done early in pregnancy, not when it's already been too late. So you get something that looks like this. So, if you look at the graph on the left, you can see over here that is, if you're plotting it against disease risk or antidepressant score, we might have the CATIM's blood test, against the other axis. And imagine if all these little colored dots are basically biomarkers that is found within yourself. Everybody's going to present with a different biomarker profile. But what you might see is that you start to see these little categories that you can start to stratify, I guess, these patients, right? Some might have a medium to low, some might have medium to high, some might have a high risk. And you can start to be able to intervene and, start to give them different treatment plans based upon social support, or maybe they might be on one antidepressant versus another. And that gives the clinicians a chance to be able to, intervene early. And if you look at it from a commercial point of view, discovery is really only the first step, right? You identify your candidate biomarkers, you do some development and some assay kind of validation to ensure that what you're looking at is something that you can measure very specifically and very accurately. And then you go through the developmental stages where you then develop this test and you get it through regulatory approval, validating this using real-world samples, and then going straight to clinical use. Now, I'm literally at like the first stage now where I looked at Olink technology, the Explore HD, in collaboration with AGRF to identify these candidate biomarkers. And really, I just want to quickly touch on this as well, because if it's not for MADA research, as well as Vicky Clifton here, as you can see, none of this work will be here today. So Vicky's actually a group leader over at MADA. She has a lab here at TRI, but her team has basically come up with the Queensland Family Cohort. And this cohort's really amazing, as you can see here, because they've tried to capture all these different areas of expertise within this study. So if you think about the samples and the questionnaires that were developed in order to capture this data, it's a lot. So it's literally a huge, massive database of not just biological samples, but all this clinical data. So one of the first things I did was to collaborate and to partner up with the Mother Research as well as with Vicky Clifton. And I said to her, look, I'm really interested in postnatal depression. I'm really interested in mental health, and I'm really interested in pregnancy. Is this something that you might be interested in? And so long story short, after about maybe 14 months of the rigorous process of going through ethics and governance, I became the first commercial company to gain access to this database, and it is really, really exciting. So you can see over here that basically at the Mother Research, they recruited about, I think it was about 500 moms in all of Queensland. So there's any mom that is due to give birth at the Mother Hospital is included. And this includes, people even with same-sex partners, with non-birthing parents, everything. And so it was really exciting to kind of see, how we could use this cohort study. Now you can see here at 12 weeks, which is the enrollment, a whole bunch of questionnaires were given up. So demographics, your medical history, medications and all of that. And then there were bloods taken at these different time points. So at 24 weeks gestation, 28 weeks, 36 weeks, and then 6 weeks postpartum. And you can see as well that mental health questionnaires were included specifically at the 24-week time point and the 6-week time point. And if you look at the bottom there in green, the clinical data, we have all this information as well: alcohol, drug use, medications that they might be on, heart rate, pregnancy complications, etc. So we have this opportunity here to really understand the biological and environmental factors that may actually impact these women. So this is what we did. So basically, we had about 439 moms that came through the study. Out of the 439, about 311 of them had completed an EPDS, so the Edinburgh Postnatal Depression Scale at 6 weeks. We identified that out of these 311 women, 36 of them were considered to be high risk. In other words, having a score of 13 or higher out of 30 on the EPDS, and women who were on a low risk. And this is about 30%, 13%. And that's in line with what we see in the literature, where in developed countries, we're seeing an average of about 11 to 12% of prevalence. But if you're looking at countries like in North America and really kind of lower kind of socioeconomic countries, that number can go up to 65%. So I was kind of glad to see that we could at least capture that statistic. Not glad for these women that have it. And one of the things that we did do as well is identify out of these women who had the EPDS completed, how many of them had bloods taken at 24 weeks. And we saw that out of them, there was about 174 women, so about 60% of them had bloods that were collected at 24 weeks. Again, the number has decreased now from 36 women to 24 women. And then we applied a very strict exclusion criteria. So we looked at all the different confounders that could affect the biomarker profiling we're doing. So anyone who had a history of mental illness, any medications they were on, any pregnancy complications, we removed all of that, and we called it the clean cohort. And we ended up with 10 out of 439, which I was still very happy with. I was like, oh, at least we got something. And so from there, we had 3 women that had a high risk and 7, that had a low risk of PPD. And then we called this the clean cohort. And then what we did is that we also considered adding some moms in there that didn't have any EPDS score. So in other words, they have their bloods, but for whatever reason, they could not do the EPDS at 6 weeks, whatever that reason may be. And so we called it the spike cohort. So we wanted to see if we spiked our analysis with these participants, could we actually see from their signature if anything's happening without actually knowing what the EPDS score was. And then we added in some real life samples, meaning we added in at random some samples, some mothers who we didn't remove any confounders. We just added them straight from the cohort, and we called them the messy cohort. Total, we had about 50 participants that we managed to put through using the Olink technology. So you can see here, our lovely Ben gave me this picture, and I was like, I need a picture for social media. And so he gave me this wonderful picture, and that's me panicking. That's my panic phase before I'm shipping off 50 samples to Melbourne, which costs a lot of money. So I was like, please don't drop these samples. And I'm really was very pleased to have the AGRF team help me with this and be able to do this for me. And so these are the results that came out. So basically, these are the results that came out from the clean cohort. Okay, so we're talking about the 10 women who basically didn't have any confounders at all. So these are what you would call de novo kind of results. So we had 3 proteins that came up as highly significant. As you can see here, this is a volcano plot. So where the 0 is, anything that's under that is basically an underexpression. Anything of over that is an overexpression. So it's just a normalized value. But what I want to say here is that when we looked at this plot, I didn't even expect to see 1, let alone 3. So I was really excited about that. But the nice thing about this as well, it comes with the Olink's platform, with the analysis platform. So again, being able to use something that isn't based upon my analysis, I'm unbiased to the analysis. This is just exactly what came out on the software. And so as Ben was showing just now, you get something that looks like that. You get a score rank, you get, the MPX difference, you get the p-value, adjusted p-value, and whether it's significant. Now, for someone like me who doesn't like numbers very much, this is like dream. I don't have to do any analysis. I literally just look at this and go, yes, I'm happy for that yes there. And so I was really glad to see this. And what they do as well, as you can see here, it automatically plots your results as well. So we're looking at just 10 here, 3 PPD, 7 controls. And again, this was exciting to me because even though it was a very small number, we were still able to see such high significance between these proteins. So that was very exciting for me. And one of the things that I tried to do as well was that I have my CSO, my Chief Scientific Officer, he's got a psychiatric background, but also is a statistical geneticist. And so I ensured, hey, I'm going to give you this data. I'm not going to tell you anything. You're going to be blinded. I want you to do the same analysis using something completely different. And he got the same results. So that made me very confident as well that, okay, we maybe have something here that's quite big. Protein B is the same. We did see this huge difference between the controls and the women with high risk of postpartum, and same with protein C. Now, you might think, okay, great, you've got some biomarkers, excellent. Is there any kind of, other analysis that we can do to look at the specificity and the accuracy? So one of the things we did then was really to look at the area under the curve. So you can see here that when we look at all 3 proteins combined, we had roughly about a 67% sensitivity. But when we looked at just protein B and C, and if you recall, these were the 2 proteins that were overexpressed and protein A was underexpressed. So if you're looking at the 2 proteins that were overexpressed, and we looked at 2 of them together, we saw that the AUC was up to about 94% accuracy. And if you compare it with the table on the side here, this was a recent study that was done that looked at the gold standard for postnatal depression and the sensitivity and the specificity there. So if you look at the cutoff value of 13 here, you're looking at a 66% sensitivity and a 95% specificity. So we're nearly or at close to that specificity there, but much, much higher sensitivity. So we're very happy and excited with that. And if you look over here, this is our heat map. So we got the protein A's, B's, and C's. And again, as I mentioned to you, the little red column here is our cases, and the blue ones are the controls. And again, with our PCA analysis, you can see how well they're grouped together. So we're really excited to see this signature here, that we can identify the ones that are clearly significant. Now, one of the things that I did do was to try and understand, okay, we got these 3 proteins. How are they related? Why are they so, it seemed quite random to me. But when I looked at it again, they actually converge from just 1 protein, and I'll call it protein X. And sorry for the A, B, C, and X's because I've got provisional patterns on these proteins. But what we identified is that these 3 proteins together seem to combine at this one single protein that when I look through the literature seemed to be a novel target that hasn't been shown in psychiatric illnesses at all. So I was really excited to see this because it's a completely different mechanism of action that I've never come across. And again, the biomarkers is also novel, looking at risk, predicting the risk of postpartum depression. So you can see here as a summary, this is kind of what I've done. I've got the 50 moms there. I've got the Edinburgh postnatal scale where they were basically stratified into high risk and low risk. And then I did my biomarker profiling. And now just from the 50 moms, or I should say the 10 participants at the end of the day, we identified 3 novel biomarkers and 1 novel drug treatment. At the moment, I've got 2 provisional patents in place and a 3rd one in progress. So what can I do with that data that I just discovered? So one of the things that I think Ben had mentioned earlier on is that the nice thing with proteins is that we can start to look at druggable pockets. And druggable pockets is one of those things where, pharma might actually be interested in looking at because A, you got a protein, but can we actually target it? Can we actually drug it, right? There's no point finding a protein if there's no drug target, nothing that can actually, attach to it. And one of the things that I did is to look at the proteins of that protein X that I mentioned to you, and that's not the protein X, but as an example, they have this little pocket, which saw these colored kind of spots there, and the druggable probability score was more than 0.9. So in other words, the protein that is novel to us has a druggable probability of about 90%. When you look at it as a diagnostic kind of tool, you can see over here that when I looked at the top 10 proteins, so not looking at just the top 3, but including the top 10, we see this kind of signature seem to remain consistent, but it wasn't significant. And I would say it's because we just don't have the numbers. 10 was a really small number. So, we're hoping to repeat this in independent birth cohorts to see if we can get this signature going as well. So one of the things that I'm really, really very grateful to all these people that's come on board to help and to partner with me, because I think, as a startup company going out into the real world, it's really hard to do these things without the collaborations that I can't thank all these people enough, as well as my team. So, I'm the CEO and founder. I've got my CSO and co-founder here, and I've got Dr. Carrie Hilliard, which is, very well known in her field as well, and is also, part of Mother. So I'm really excited to have these people on board my team as advisors as well. So with that, I really just want to thank the participants that participated in the QFC study, more so the Mother ethics and governance people who have spent a lot of time helping me with this application. Vicky, of course, who's really the champion of CATIM, and a lot of all these other people who've really put in that help that I needed when I first launched the company. Thank you so much.